Welcome. In this class, we would look at another application of transport. Okay. Especially in these two classes, the last two classes of this kind are going to look at the application of pseudo-steady state principle in our research. Okay. And that has led to so much insight, so much benefit in terms of the knowledge generated for improving bio-oil productivity on one side, and even dealing with cancer or managing cancer, treating cancer, those aspects, or for that matter managing any disease. This insight is going to be helpful. Okay. So let me first talk about the bio-oil aspect. Sometime in the middle of the previous decade, or let me say 2004, 2005, the oil prices shot up and the interest in the alternative energy aspects came up again. It typically happens in cycles, and there was a lot of interest in bio-oil from microalgae, biodiesel from microalgae, bio-oil from microalgae, and so on and so forth. Let me show you how the principle of pseudo-steady state was effectively used for the system, okay, in our study. To give you a little bit of background in terms of molecular aspects, you all know vegetable oils, right? Vegetable oils are the ones that we use for cooking and so on and so forth. Where do they come from? They come from vegetables. Why? What aspect of the vegetables leads to oil? They are the lipids in those vegetables that are oil, that are extracted out by pressing them and so on and so forth, and they are used. Similarly, you could get oils from microalgae, right? Same way, same kind of molecules that contribute to oil from vegetable sources, similar molecules contribute to oil from microalgae sources. The crude oil that we get out of the earth has a different composition, but at certain levels it can contribute to these fractions also. You get a wide variety of fractions there, and the difficulty is with respect to crude oil and the pollution, the carbon dioxide, it's a greenhouse gas. There's a lot of climate change aspects that are caused by a heavy use and indiscriminate use of crude oil. Essentially, you're taking out carbon that was formed millions of years ago and putting it out into the atmosphere and so on and so forth. Major difficulties which were viral in about 15 years ago. So all these things came in, and we also started looking at it from the reactive species angle, which happens to be our research core line, if you want to call it that. The idea here is something like this: microalgae, bio-oil from microalgae, and reactive species related aspects. We already know that the stresses cause reactive species in the cell, reactive oxygen species, reactive nitrogen species, reactive halogen or chloride species, and so on and so forth, right? We of course focus on hydroxyl and superoxide here, which are ROS, and we proposed and used the specific or the per cell levels of hydroxyl and superoxide as quantitative markers of stress, irrespective of the means that were used to cause the stress. Okay. And this is where the pseudo-steady state comes in. Why? Because the kinetics of these reactive species is very fast. Hydroxyl radicals 10 power 10 per molar per second, right? The superoxide radical anywhere from 10 power 3 onwards, higher, and so on and so forth. So when you are interested in processes related to growth, which happen over time scales of about an hour or more, and so on and so forth, doubling, right? The reactive species reactions can be considered to be at steady state. Okay. In other words, we don't worry about the unsteady nature associated with the reactive oxygen species reactions when the interest is in the much slower process, which is growth rate. If you do that, then it becomes then it provides you with significant insight, and you could actually use the pseudo steady state levels of hydroxyl and superoxide as quantitative markers of stress. Okay. And we showed that it was they are fundamental mediators of stress, irrespective of the means that are used to cause stress. This paper and other papers, irrespective of the means that are used to cause stress, they are caused by the imbalance between the production and destruction of the reactive oxygen species in the cell, or the amounts accumulated lead to stress. So, by using the pseudo steady state principle, we had a means by which we could address these aspects quantitatively. So, we saw two conditions that were perceived to cause stress. Okay, let me switch to the paper at this stage. This was again published in Biotechnology and Bioengineering in 2013. Stress induced lipid production in Chlorella vulgaris, relationship with specific intracellular reactive species levels, fundamental mediators of stress. Okay, that is what we established here. Kavya Menon was my MS student, Ranjani Balan my PhD student, and myself. And I think I will read the, is it here? Yeah, it is here. So, I will read the introduction. The abstract tends to be a little too focused for an undergraduate course. Okay. Attainment of sufficient cell concentrations and sufficient specific lipid accumulation levels seem to be two major challenges in the commercialization of algae cultivations for bio-oil production. Stresses can be used to improve bio-oil productivity in microalgae, and many studies on the effects of stresses on algae, such as UV treatment, temperature, pH, salinity, nitrogen deficient growth media, have been reported. Other stresses such as light, nutrients, symbiotic relations have been found to induce changes in the cellular composition and so on and so forth, but have not been exploited well toward improved cell yields. Although the effects of many stresses have been investigated, the understanding of their effects seems to be fragmented, possibly because a unifying view has not been employed. Okay, this is where we contributed. We contributed a unifying view in terms of the reactive oxygen species. The reactive oxygen species are known as the known to be signaling molecules for various cellular events, and it is reasonably well accepted that ROS are the molecular mediators of various stresses. We propose to use the specific intracellular reactive oxygen species level as a quantitative marker for the stress, irrespective of the means used to induce the stress, especially because easy quantifications of SIROS are possible through fluorimetry. It is known that SIROS levels are higher under stress, although only a few studies have quantified such levels. Our lab is one such lab, few labs that quantifies it. Even if the studies that quantify ROS levels, the common quantification is of total ROS that includes many species such as superoxide anion radical, hydroxyl radicals, hydrogen peroxide, hypochlorous acid, peroxyl, alkoxyl, singlet oxygen, and peroxynitrite. And all these are measured by something called DCFDA. Okay, it measures some mix of these species. In our lab, we measured the specific levels of individual reactive oxygen species, say hydroxyl alone or superoxide alone, separately, which we believe would facilitate a better understanding of the various roles of ROS and their effects on many different aspects of cell function. One of the perceptions that seem to discourage, okay, this is where we come to transport directly. One of the perceptions that seem to discourage the measurements of the actual amounts of ROS, either as concentrations or as specific levels in the culture, is that the species are short-lived and hence may not contribute significantly to the information on cell status. Nevertheless, we have found in our earlier studies in our lab that actual amounts do provide significantly useful and quantitative information. This dilemma could be resolved when the rates of the various relevant processes are considered. It can be seen that the fast rates associated with ROS reactions makes it possible to consider them as being a pseudo-steady state in comparison with the much slower metabolic processes or growth. Okay. This is the crux here. When viewed in the context of pseudo-steady state, the net specific ROS levels can be considered as an indicator of cell status or the stress levels in the cells at a particular time point. Okay. Then this, okay, let me read this and then summarize. This work addresses the effect of stresses induced by nutrient and light on microalgal growth and lipid production, nutrient levels and light wavelength, higher frequencies and hence higher energies are perceived to cause stress in cells. In addition to the investigation, we did something else also. Okay. So, these are the materials and methods, these are the results and discussion in a certain framework which may not be very relevant for our discussion here. Yeah, what I do is I would go directly. You could read this if you have an interest. Certainly, it's a very interesting set of results that we have here. I will go to the summary directly. Okay, that might be a little better. We looked at two conditions that are perceived to cause stress. One is different nutrient levels and different light frequencies or wavelengths. Okay. The model system was Chlorella vulgaris, a microalga, and these were the results. You see cell concentration on the y-axis, cells per ml, okay, versus specific intracellular hydroxyl radical level, okay, in moles of hydroxyl per cell. Okay. You see here, this is the various nutrient levels 1x, 2x, 4x and so on and so forth compared to a certain control. And this corresponds to the data with blue light, this corresponds to the data with red light, and this corresponds to the data with white light. Okay. All this data of cell concentration versus hydroxyl radical levels nicely falls in this region. Okay. It's a beautiful relationship, okay, which shows the fundamental nature of the effect of hydroxyl radical levels. Higher the hydroxyl radicals, lower the cell concentrations. This is irrespective of even the various phases of the batch growth that we went to under these conditions. All of them nicely line up here. Okay. And here you have cell concentration versus specific intracellular superoxide radicals. This is hydroxyl radical, highly reactive and so on. This is superoxide, highly reactive, as well as it plays signaling roles in this cell. You could see as these levels increase, the cell concentration goes down. Okay, beautiful relationship, and this was actually quantified also as see, this is a log scale, right? So y equals k1 times x bar minus 0.7 was the line that corresponds to this equation corresponding to this line, and y equals k2 times x to the power of minus 0.79 is the equation that corresponds to this line. And more interestingly, this is the effect on specific intracellular neutral lipid level in the cell, which is nothing but the one that is oil. Okay, the neutral lipid is the one that will give you oil. It was measured in some terms and so on and so forth. Don't worry too much about this. So this is specific intracellular neutral lipid level. This is specific intracellular hydroxyl radical level. Beautiful linear relationship with all the data put together, and linear relationship. This was k3 z equals some z equals k3 times x bar 0.65. Okay, z axis, x axis, and so on. So this was actually published as a cover article with video abstract. Video abstracts were considered prestigious at that time when it was first introduced, and it was published in 2013 in Biotechnology and Bioengineering. Okay. Welcome to this abstract. We all know that bio oil from microalgae is an area of current research interest. The process of making bio-oil from microalgae is quite simple to describe. Nevertheless, there are no commercial plants across the world that utilize this process to produce bio-oil from microalgae. That is essentially because there are very many challenges to this particular process. If we focus on the upstream side, we can consider getting sufficient cell concentrations as well as getting each cell to have a sufficient amount of bio-oil in it as two major challenges. In this particular work, Kavya Menon, Ranjani Balan and I have looked at some aspects of this. Now let us look at stress. People have considered various kinds of stresses to improve the yield of bio-oil or the productivity of bio-oil from microalgae. When we think of stress, the first thing that comes to mind is its relationship to the reactive species. In this work, we focus on the reactive oxygen species and especially the hydroxyl species and the superoxide species. In fact, we propose to use the specific level of these reactive species or the amount per cell as quantitative markers of stress, irrespective of the means that are used to create this stress. And one of the first questions that comes to mind is the reactive species react very fast. So, if the reactions are so fast, how can they be considered as markers of something? Nevertheless, we have found that these have provided us very useful quantitative information in our studies done earlier, and this difficulty can be resolved by considering the pseudo steady state aspects. And since these reactions are so fast, you can consider them to be at pseudo steady state levels in comparison with the kinetics of slower processes such as metabolic processes or growth processes. In this work, we have considered two conditions that are perceived to cause stress. One is different nutrient level and also different frequencies or wavelengths of light causing stress. The model system in this particular study is Chlorella vulgaris. Cell concentration was a specific hydroxyl and specific superoxide reasonably linear. Something interesting happens in the case of neutral lipid levels too. Let us just look at neutral lipid levels versus specific hydroxyl levels. When all the data are put on the same graph, there is a relationship that we see, and there are other interesting results in the manuscript. The findings provide a step for us to rationally design the stress based strategies for improving bio-oil yields from microalgae possibly, and personally there are more interesting results, such as questioning what exactly stress is, the very notion of stress. Kavya, Ranjani and I warmly invite you to read the paper. In fact, we have made special efforts to make the paper easily readable. The title is Stress Induced Lipid Production in Chlorella vulgaris Relationship with Specific Intracellular Reactive Species Levels, and I need to acknowledge the funding that was provided by the Department of Biotechnology, Government of India, the Department of Science and Technology, Government of India, and the Divashri Fellowship by Srikumar Suryanarayan, a distinguished alumnus of IIT Madras. Happy reading.